It was Christmas Day, December 25th, 1900. When the ornithologist Frank M. Chapman took hold of the burgeoning conservation movement in North America. Instead of hunting birds as part of normal holiday traditions, Thaddeus Chapman decided to count them instead. The Christmas Bird Count, or CBC, had humble beginnings, starting with 27 dedicated birders, but today has evolved into tens of thousands of volunteers all across North America every year. This annual census of bird species has directly contributed to conservation efforts led by the Environmental Protection Agency and climate change reporting. What does bird counting in North America have to do with the things we normally talk about on this channel? If it's your first time here, I'm Jack Wang, a microbiologist and science educator based in Australia. The Christmas Bird Count is one of the earliest examples of citizen science, a template for large-scale scientific participation involving members of the general public that is gaining traction across all areas of science. Today, let's walk through how teachers can use citizen science to promote active inquiry-based learning. At its core, citizen science has three basic ingredients: scalability, authenticity, and communicability. Projects that rely on a large number of people for success is not a bug, but a feature of citizen science, and often the pool of participants extends into the general public. This brings with it varying levels of scientific knowledge and training, but what the participants do still has to be authentic research with the potential to discover novel outcomes. Innovation is the driving force for participant recruitment, and so there needs to be a hook. Why should people care about this project, and how does it impact their everyday lives? This of course relates to the communicability of the project and how easy it can cut through the noise. Seeing as citizen science projects cannot happen without the participants, researchers have to communicate the findings back to the broader general public as well. It's hard to consider scalability and communicability without first establishing the authenticity of the research project. In our field of molecular biosciences, authentic research revolves around the lab, where each experiment uses expensive reagents, complex equipment, and interconnected multi-step protocols. We can't expect non-scientists to start contributing straight away. There needs to be support mechanisms in place to slowly ramp up the complexity. One evidence-based way of doing this is broadly termed inquiry-based learning, where students are challenged with open-ended questions in lab classes, the answers to which are unknown to them. These questions may not be innovative or interesting to a broader audience, but students need to use problem-solving skills as part of their learning. Inquiry-based learning can take place across a continuum of student responsibility, ranging from verifying the results of famous old experiments by following a recipe-style protocol a la a cookbook, all the way to an immersive apprenticeship-style research experience. Inquiry-based learning has been shown to improve student performance, processing skills, and attitudes towards science, and these benefits are further amplified if teachers give students even more autonomy through research-based experiences. We're talking more confidence and enthusiasm towards research, higher learning gains in analytical skills, and more advanced experimental planning. That's all good in theory, but how does it work in practice and become a citizen science project? The first step is to convert one of your existing lab classes into an inquiry-driven format. Let's use an example from one of my microbiology classes. Classic Introductory Microbiology asks students to swab different surfaces in the environment or on their body to isolate microbes in broth cultures or agar plates. Often it's also used as a tactic to scare junior doctors into washing their hands more frequently. It's not particularly innovative though, and with a few simple tweaks, we can make it more inquiry-driven. Instead of prescribing students specific places to swab, give them a choice. Say pick 3 out of 10 different body sites or different environmental samples and ask them to make a prediction or hypothesis. Which sample will have the most amount of growth or the most diverse range of microbes? And how will they test these hypotheses? You can provide them with the exact techniques to use, but this is also too prescriptive. Students can be given standard operating protocols though, and it's up to them to choose which technique will work best. They could do a serial dilution of bacterial broth and count colonies on plates. They can measure the turbidity through OD readings on a spectrophotometer, or use light microscopy to do cell counting assays. Each of these have their pros and cons, of course, and the learning happens when students have to cognitively process which is better suited for their own experiment. It's up to them to figure this out. What if your labs are only set up to do a small number of techniques in a limited amount of space? It's actually fine to focus just on one technique and bring in inquiry elements through optimizing the protocol. Does it matter if the swab was dipped in saline first or not? If you use chemicals, what concentrations work best? Can they perform a serial dilution of the chemical to figure this out? Optimizing and adapting existing protocols is a huge part of science, and there's no reason why it can't be a bigger part of science education. Now, Now that we have embedded more elements of inquiry, how do we upscale this to citizen science? Every teacher wants smaller class sizes to better connect and relate to each student, but in Australian universities, we're often dealing with hundreds of students in each class. It's a challenging environment, but citizen science is uniquely positioned to take advantage of this. Instead of a broad call out to the general public, you can simply pitch the project to your own students and still have a big sample size of participants. Using my microbiology class again as the example, we crowdsourced student volunteers, up to 400 students a semester, to determine the microbial variability across human populations, the Human Microbiome Project. We've all seen ads that claim the latest yogurt is full of only good bacteria, but what counts as good or bad bacteria is actually a pretty open question in our field. Every student was already swabbing their body sites to culture microbes anyway, so we simply shifted the context of this work towards a global research priority in our field, with all the ethics approval in place, of course. Students swab their own oral cavities and are given a suite of personalized diagnostic tests to identify the microbial composition within their mouth. The human microbiome has correlations to chronic diseases such as asthma, diabetes, and even cancer, and students contributed their de-identified biometric data towards the research of novel therapies. My teaching team was actually one of the first in the field to provide thousands of students with personalized next-generation DNA sequencing technology to characterize their own microbiomes, and we published our findings in a microbiology education journal. This ties into the last piece of the citizen science puzzle: communicability. The findings need to be communicated back to the general public, but it doesn't have to be in the form of a peer-reviewed journal article. It can be more immediate in the form of blogs, podcasts, or videos, and you can even make this part of students' assessment. For example, I asked my students to form pairs and record a podcast discussing the findings of the microbiome project for the general public. Communication works best if the medium is tailored to the target audience. We've talked about this in a previous video, and online content is how most people find out about everything these days, including science. It's very tricky to do this well, and we've had to adapt the marking criteria for the podcast assignment over the years to focus on things like lack of jargon and use of metaphors or analogies. It's been a really fun process actually, but perhaps more on this in a future video. Who would have thought that counting birds on Christmas Day over 100 years ago could serve as a catalyst for something like the Human Microbiome Project? It just goes to illustrate a potential for science to go in any direction imaginable. And there are hundreds of recent examples of citizen science in my field alone. Over the next few videos, I'll spend some time walking through how specific lab techniques can be adapted towards inquiry-based learning and citizen science research. This is the Biolab Collective. I'm Jack Wang, and I'll see you in the next video.